Okay, so I think we will just start the session right now. So I'm trying to share the slides. Just let me know whether it's clearly visible and whether I'm audible or not. If it's working fine. So are the slides clearly visible? Somebody just lets me know.
Yes, sir. It's
Okay,
visible.
fine. When I thought I lost my connection for a second. Okay. Yeah, so again, we will continue with the week 8 live tutorial session. So this is the course, Bioinformatics Algorithms and Applications, instructed by Professor Michael Bromiha. In the session of week 8, could you tell me what Professor Bromiha went over briefly in his lecture, uploaded lecture videos?
In week 8 lecture, Professor Michael Gromeha go through in detail about the database of Protherm and how can we use the thermodynamic relations such as delta G and energy potentials and all, and to differentiate between the mutant result and the uh, experimental result. So he got into detail about how to build up a database such as a thermodynamic database in throw such as such as Prothrum.
Yeah. So different databases, concepts related to mutations as well, right? Stabilizing mutations, destabilizing mutations, this kind of
Yes, sir. Yes,
yeah,
sir.
and
Yeah.
some thermodynamic parameters and how we can interpret that. So we'll try to go through it. First, I guess we will again just brief about what we talked about in the earlier classes. And then we'll try to solve some questions as well, some questions that I did not have the chance to go through in the last week's class. So yeah, okay, we'll just go forward with that. Yeah, so what we just to briefly cover what we did in the last class is that we talked about some concepts, for example, contact map, right? So contact map, these can give us an idea about um, basically if certain residues are forming long range or short range contacts, so on and so forth. So for example, if particular residues are within range in a 3D space, so that means they should be forming some sort of context, right? Now you can define any sort of cutoff of distance so that you can say that if the distance between these particular residues is within a certain range, so they are forming contacts, right? So this can explain a variety of interactions. For example, you might find out that they are forming whether hydrogen bond, right? Pi-pi interaction, cation-pi, charge-charge Coulombic interactions, kind of salt bridge-like formation, right? So different interactions are possible, hydrophobic interactions are possible, but based on just the distance, you can establish a cutoff, and therefore you can find out whether they're forming contacts or not. Now, of course, if residues are close by, so that means that they will form some sort of contact, right? It's expected that their side chains orientations will also kind of be close by. So those residues where they are basically performing contact and they are also within a very close range in the sequence sort of space or sequence information. For example, let's say residue 4 and residue 6, right? Or residue number 7 and residue number 10, right? So you can say they are forming short range contact because they are present very close by in the sequence space. They are also forming contact in the 3D space, right? However, what happens is that since the protein folding is very complex, right? So what can happen is that it can fold in such a way that much distant residues in the sequence space, they can also come together and form certain type of contacts, right? For example, let's see, we're talking about let's say one lysine residue was here at let's say 40th position, right, in the sequence, and then one aspartic acid, let's say it was there in the 88th position or 85th position, right? So there, of course, 45 45 amino acids are part in the sequence. However, the folding could be in such a way that in the 3D kind of conformation, they are coming together and they are sort of forming a salt bridge interaction, the lysine 40 and aspartate 85. So that means that this is the example of a long range contact, and the long range contact would be very useful because these type of interactions are what stabilizes the um, you can say 3D fold. You can also say certain type of structural kind of motif or certain type of fold of a particular motif. It could also be the fold of secondary structure, so on and so forth. So it has importance of stabilizing various parts of the structure basically. So that's why it's sometimes important that if you means if you look at not only the short range context but also the long range context and whatever interactions they are forming basically, right? So for example, here we can see that arginine 6 and isoleucine 7, those two are situated very closely together here, and that's how we can say that they are forming a short range contact. Similarly, in this particular contact map, so this was what I'm talking about. Similarly, in this particular contact map, if you see that, let's say the residue 2 and residue 10, right, they are also forming contact. So if your cutoff is below this particular range, you can also say that these two are forming kind of like a long range contact, right? So that's what we went over. We went over briefly on the classification of protein based on the structural prominence of what type of secondary structure we have. For example, all alpha type of proteins will mostly be dominant in alpha helices. All beta type of proteins will be dominant in beta strands, right? And then we also looked at alpha by beta and alpha plus beta type of structures, where basically your alpha by beta or alpha slash beta proteins, they will not have segregation. So that means probably one beta strand and one alpha helices are kind of alternating in the occurrence in the secondary structural formation, right? Whereas in the alpha plus beta protein, you will see that there is some sort of alternating pattern between beta strands and alpha helices, but multiple beta strands and multiple alpha helices will be present at a single time. So that means that there is some sort of segregation is going on. Similar type of secondary structures are clumping together, even if there is an alternating pattern between the different ones, right? So that was alpha plus beta proteins in this case, right? So next we went on to cover some of the informations, for example, center of mass, right? Center of mass. This is also related to your PDB structures, right? So PDB structures are what uh, contain the coordinate informations that are deposited from X-ray crystallographic experiments or cryo experiments and such kind of experimental procedures. And then during different sort of analysis, you of course have to calculate multiple parameters, right? And many of those parameters will be based on the center of mass. For example, a distance, right? If you want to calculate the distance of let's say one particular motif to another particular motif, uh, then what you cannot take a single point or a single atom as a particular point to carry out these distance calculations, right? So in that case, what you have to do, you have to calculate the center of mass of this bulk motif overall, and then you have to calculate the center of mass of the other one. And then from those 2 points, you can calculate the distances. So that's why center of mass is also quite important, which can be found out by this formula. So the coordinate of center of mass particularly, that is quite important, so that you can establish a particular point where uh, your different calculations can be useful from basically taking those particular coordinates as a reference, right? And then the biggest one is that we went over something called homology modeling. So homology modeling, also called comparative modeling. So it's a process whereby if you have a unknown structure, if you do not know the structure of a particular protein, but you have the sequence of it, and then if you know the sequence of some other protein, which is kind of homologous to this unknown one, and then you also have a structural information for that particular protein, right? So from that, what you can do is you can build the structure of the unknown protein based on their sequence and alignments and so on, right? So basically the process was that you first you have your target sequence, then you try to do a database search, try to find out a good template, then you do a sequence alignment, you do refinement of the sequence alignment, and for from that particular, you try to first build the backbone, and then you add the loops at the side chain, the model optimization, and finally comes the model evaluation. So does anybody remember how did we build the backbones particularly? So you have the sequence alignment, right, of your target and your template protein. So how can you build the backbone structure of the target protein?
Sir, if the sequence alignment is very high, we can just copy paste the backbone because it's the same. Because it's just carbon and nitrogen atoms and have peptide bonds. There is a little bit difference between the some amino acids are changed, so we can just copy paste it.
Yes, yes. So for backbone, you can particularly take the backbone atoms, which are your N, C, C alpha, and O. So you can take that from the template protein, and you can copy paste that to the target protein. Uh, and let's say if you had not only similar but the exact same amino acid residue in a particular alignment, then what you could do is that you could also copy and paste the coordinates of the side chain atoms for that particular amino acid, right? So you could do that. Otherwise, for building your alignments, you can also uh, take into account the different rotamer libraries, right, which will be important for sidechain sort of sidechain building, right, sidechain building and modification uh, calculation in this case, right? And then for the loop modeling, which we went over, uh, so it can be done via database searching as well as in ab initio method, right? So in the ab initio method, what was that? Basically, it was generation of definitely a large number of random loops, and then from there, profiling of their phi angles, which ultimately will be the factor for deciding which loop conformations are possible, right? To define phi angles, as well as let's say the algorithm that has information for the positioning of the environment, because all of the other residues are kind of built. So whatever positional restraint needs to be there, so that information is already there. Now, if you sample the different phi angles and find out the optimal ones, so you can build the loop in that way also, right? So different integration of this phi profile as well as force fields, etc. So they are involved in this sort of loop modeling process. We also went over one example using the modeler software, right? You can also do the same via the Swiss model software, which is again an online tool. So rather than installing modeler, you can also go for the web servers basically, or web based tools, right? So developing web based tools is also a big part of bioinformatics. So you can take a look into that as well. Of course, following that, what we have to do is that you have to do the refinement of the model, right? Now, even if you, let's say, build a model, you use different algorithms and you try to just do the loop modeling, sidechain modeling, etc. You will sometimes see that not all of the steric hindrances, steric interactions that had occurred, they are alleviated, right? So for that, again, because the algorithms definitely for the most part, they also take into account the resource effectiveness, right? So it's not that the algorithm is running for a long time. So it's running and then it's trying to see a convergence point, and from that it is deciding that okay, this conformation is possible or that one is not possible, so on and so forth, right? However, those are in the interest of like computational resources, they definitely do cut some corners, right? So it's not always that a single algorithm or a small scale calculation will give you the exact conformation. For that, you will have to optimize sometimes energy minimization, etc. For those conformations, right? So, for example, here it's talking about a couple of hundred of iterations of energy minimization. For example, different types of calculations you could run. Let's say like steepest descent, conjugate gradient, these are some types of minimization algorithms. So you could run those type of algorithms. And then it is also talking about that molecular dynamics is also a commonly used process to refine the structure, right? So molecular dynamics is a kind of a big process. So usually when you run molecular dynamics, before that you have to do some sort of energy minimization. You have to do the heating of the system algorithmically, of course, to the temperature that you want to explode your conformation at, for example, at 300 Kelvin. And then you have to slowly relax the system as well. So that is through like some sort of equilibration run, and then probably you will do a production simulation, right? So through the production simulation, you will get the different conformations that are accessible for this particular protein. If the force field, etc., are good enough, then you should get basically good structure if you want to do any sort of further calculation or computation in that case. Okay. So that was model refinement, etc. And then model evaluation, of course, you have to do check the different phi-psi angles, phi-psi profiles using Ramachandran plots, whether those are in good agreement to the standard or not. There are some tools, softwares, for example, Project, where you can do it. And then after the homology modeling discussion, we went over CASP, right? CASP was the critical assessment of protein structure prediction. We talked about this yearly competition where AlphaFold also shown through. I guess the major success was in CASP 16.
2020, I mean,
Yeah, okay.
18 to 19.
Yeah, no, it's written here CASP 14, right? 2020. Yes. So in CASP 14, I believe they surpassed the score of 90 that the organizers had already set, right? So after AlphaFold got recognition, of course, you know that the developers also got Nobel Prize, Demis Hassabis and John Jumper. And we briefly talked about Professor Baker's lab also. They work on computational protein design. So basically trying to uh, design some sort of structures, protein structures that do not exist in nature, but you can tailor them to particular type of functions. For example, in sustainability, like in the case like just as an example, let's say plastic degrading enzyme, if that is possible, right? Or let's say snake venom, some sort of protein that tries to combat those, right? So these sort of will be useful in different parts of the world if the technology becomes feasible. So his lab works on this kind of design. Then they also validate them experimentally. So very good work that they do there, and of course they have been recognized for the same. Yeah. Okay. So yeah, so Professor Gomiha in the lecture, he went over structure comparison with something called DALI, DALI, right? So do you know what the particular method that particular principle that the software uses? Can answer in chat. Yeah, I see you talked, you answered contact number C, right? Contact map. Yes, exactly. So this particular tool, they use contact map for comparison, right? So here is kind of the link of the server. So it is talking about conservation mapping in 3D, right? So it's a program for protein structure prediction, as they have mentioned here. There are, of course, different parts of it, right? The processes that they follow, they have also included here. But mainly, what they do is that they uh, the comparison, etc., that they do in this particular tool, that is done by a contact map, right? So yeah. So if you see here, it says that contact map based methods such as DALI, right? So so it's based on contact map particularly. Okay. So that was correct. We actually looked into the urea denaturation experiment questions, I believe. So earlier what we did was that in urea denaturation experiments, we took into account the fraction of folded as well as fraction of unfolded proteins value, right? And then from there we could calculate some sort of free energy change, right? From a particular in a particular state, let's say an intermediate state that was between the fully folded versus the fully unfolded, right? In between somewhere around somewhere in the range. So in this particular question, it's kind of the same. So it's asking that if 4.5 molar urea, if you introduce it leads to a state where 30% of the proteins are present in an unfolded state at 300 Kelvin. So what is the free energy change from the initial folded state, right? So of course, we know any sort of free energy change calculations, it's minus RT, L, and K. And in this particular case, K will be some sort of fraction, right? Since it's talking about 30% of the protein, so we can, of course, say that it is 0.3, right? So if you look at the formula, so the fraction of unfolded, that would be 0.3. And then the fraction of folded, which is in this particular case, this term, so that will be, of course, 0.7. So you can just calculate it using this formula. So minus 1.987 times 300 times ln of 0.3 by 0.7. So could you tell me what the answer should be in this particular one? And for these types of questions, always look into what the units are uh, given, right? So if you do the calculation in calorie per mole, then you have to convert it. The options are given in kcal per mole, so you might have to convert that to kcal per mole as well. And there could be some sort of confusing, options to choose from between, like these calorie per mole or kcal per mole, and these kinds of way, because they look very similar, but to look at it, they're different values, right? So in this case, we can find a value of 505.07 kcal per mole. So remember this formula. From this one, I'll just say that remember this del G equals to minus RTL in the formula. Quite handy in a lot of scenarios, right? Okay. So we know, and we also looked into it earlier, right? The PDB structures, for example, they can the coordinates of particular atoms, they can be used to calculate the differences, right? And as I told earlier, uh, certain range of distances, they can be useful to assess whether certain type of linkages are forming or not, right? For example, if you have a lysine and if you have a if you have a glutamic acid, so if you want to know whether they are forming salt-bridge interactions or not, usually a 4 angstrom range between the residues, particular atoms of those residues. For example, I believe lysine, oftentimes used on is the NZ atom, right, in PDBs, in PDB structures, and then for glutamic acid, it will be some sort of carbon atoms, right? I think it's CD or CG. But yes, but the distance between those, if that is within the range of certain value, for example, in 4 angstrom, let's say, so they're forming charge-charge interactions or strong charge-charge interactions or salt-bridge type of formation, right? Similarly, of course, we know that the cysteines, they form disulfide linkages, right? So disulfide linkages will be the distances between different sulfur atoms, right? Sulfur atoms that are present in the cysteines. So what this question is giving you is that it's giving you the a small portion of the entire PDB structure, mentioning the different sulfur atoms, the different SG atoms of different cysteine residues that are present in this PDB, right? So for example, this is cis 23, this is cysteine 97, this is 138, 192, and 296. And you have the information related to those given here, right? Now, based on this, you have to calculate multiple distances, kind of, and then you have to identify which sort of cysteines they are forming disulfide linkages within one another. So, try to calculate the distances, and we can see it just in a bit. Okay. So, remember that for disulfide bond, again, the distance, of course, has to be very low, right? And there is some conflict between different algorithms. For example, what the cutoff of distance for disulfide bond should be. But let's say around 2 angstrom, 2.2, 2.3, you can use that, okay, for a distance cutoff for disulfide bond. So just try to calculate some distances here using this formula. So it's very simple, just root over of x2 minus x1 square plus y2 minus y1 square plus z2 minus z1 square. So you should get into it. typically your sulfur distance will be around 2.03, but certain algorithms have um, very close to that, but it could be certain different cutoffs for disulfide linkage formation, but we'll just take it around that. Okay. So I can do the calculation, we'll just see it in. Okay. Yeah, so what you basically had to do in this case is that you had to calculate the different distances, right? Uh, and from here, as you have seen in earlier PDB structures also, so this particular value here, just after the residue identifier, so that is your x coordinate, right? And this particular one is your y coordinate value, and then this particular one is your z coordinate value, right? So if that is the case here, so if you do the calculation between these different sets of residues, right, you will find out that the 23 and 97, these two residues are standing or positioned at around 2.046 angstrom from one another, and then 138 and 192, these two are positioned at around 2.051 angstrom distance from one another, right? So in this particular case, you can say that these sulfur atoms are within a disulfide bond formation range. So that means that 16, 23, 97, that will be one pair of disulfide bond forming residues, and then 16, 138 and 192, those will be another pair. Okay. Again, you can take a look at the coordinates and can probably tell which ones are in close proximity to the other. For example, if you look at 23 and 97, you will see that their x, y, and z coordinates are pretty close by, right? If you compare with 138 and 192, you will again see that the x, y, and z coordinates are pretty close by. For example, 12 versus 13, 19 versus 17, -2 versus -3, right? If you, for example, compare, let's say 23, if you compare this with 236, you will see that their x, y, and z coordinates are so far apart. For example, -12 and -21, -20, 14 versus 20, -23, etc. So it's possible that, of course, these pairs will not form disulfide bond because their coordinate, from the coordinate information, just visually speaking, they seem very distant, right? So you can just take a look at the visual cues as well to find out which particular residue pairs you want to calculate the distances for first, and then you can go from there. In this case, only 2397 and 138, 192 form the disulfide bond linkages, right? So in the question, I guess you might not see something like that, calculate these multiple distance pairs and then tell what bond is forming. It might very well be, let's say, a yes or no type of question, or kind of like a statement type of question, that whether these particular residues are forming this particular interaction or not, right? And from that you have to calculate the distance. Okay, so remember this distance calculating formula. If you are given something like this, so it's root over x2 minus x1 square plus y2 minus y1 square plus z2 minus z1 square. Okay. So in the last class also we talked about the parameter that is used to assess conformational deviation from a reference structure. Anybody remembers what that was? So conformational deviation, exactly how we could calculate? It was basically using one particular parameter called root mean square deviation, right? So root mean square deviation is something like this, where you have a for atom I, you have a target kind of coordinate position, right? And if you have the, for the same particular atom, if you have the reference position as well, then what you can do, you can subtract this particular values, you can do a square of that, and then you can divide that by the total number of atoms in this particular case. Okay, so that will be non-mass weighted calculation of RMST, and following a summation, and then following a division by the total number of atoms, you can get the value of the RMST. Okay, so the RMST will overall tell you about the deviation between the reference structure or reference atom or reference particle coordinates with the target one, what you are trying to do the calculation for. Now, could you tell me what could be some, so just based on this formula of how this RMST is calculated, could you tell me what could be the downside of RMST? I mean, as downside, I mean what kind of information it cannot capture? So it's root mean square deviation, right? And based on the formula, does anything come to mind of what could be the limitation of RMST, or what kind of information it might not be able to capture in a good way?
Some kind of ligand binding, if there is a
Uh,
ligand, like a
yeah,
cofactor or
right.
something.
So yeah, so RMST is basically kind of a deviation metric, right? So for ligand binding, you can calculate the change after ligand binding, for example, in protein, or you can compare
Yes.
the initial structure of the ligand, and then you can find out how the next structure of the ligand, or in a simulated environment, the structure of ligand, how that is changing. Okay. But the thing is that, so from a reference structure, let's say, if your let's say we are talking about some sort of loop, right? So let's say a loop was positioned in this particular region of the protein. In this particular position, your loop was initially for a protein, right? Now let's say you have run a simulation, and then you are seeing different changes throughout the simulation, of course, because loops tend to fluctuate, right? Now let's say you are seeing in one particular conformation that the loop was kind of extending in this particular way. Okay, and let's say this is the inner part of the protein, or the inner or hydrophobic or kind of sort of a bodied environment of the protein, and then let's say you are seeing that in another conformation it was like extended in this particular region. Okay, now what is going on here is that uh, for both of these, if let's say the deviation was kind of the same, so for both of these, in this one and in this one also, you will see the same RMST with the reference structure, with respect to the reference structure. Why? Because you have already squared the values, right? you have taken the coordinates, but you have squared the values, and then you have done a square root at the end, right? So that means that whether the loop conformation shifted towards the left or whether the loop conformation shifted towards the right, it does not matter. It will give you the same RMSD with respect to the reference structure, initial reference structure, kind of similar, right? So that means that the directional um, sort of information in this case that the RMSD is not able to capture. So basically multiple conformations that you are seeing in your particular protein, it is possible that the conformations are different, but it is calculated in such a way that their RMSD is same. So even if your RMSD is getting leveled up or it is staying in a particular range, it could be that the conformations are quite much shifting, right? Similarly, if you calculate, let's say, different conformations, so they could have the same RMSD also. So the same RMSD could have different conformations in the simulation, and then the different RMSD, it is possible that they are deviated in such a way that the conformations are kind of similar, right? So it's possible. So that is kind of the downside of, or sort of the limitation of RMSD in this case. So that's why it's better that you alongside these type of calculations, you take into account some sort of other metrics also. Metric that could more clearly give you the variance or the variable part of your data, basically, right? For example, you could take a look into some sort of distance-based calculation alongside RMSD. So if you had calculated, let's say, if your initial positioning was this, your two different conformations, they were kind of like this. So you could have calculated some sort of distance from, let's say, a particular conserved region, which is this highlighted, right? So you could have calculated this distance, the different distances that had occurred, center of mass distances for different conformations. And then based on that, based on RMSD and then based on the distance, you could uh, perhaps calculate a free energy distribution. Okay, so this free energy distribution will tell you that where your minima positions are forming, what is the energy barrier between those, so on and so forth, right? So that is a good idea sometimes, that take note of multiple metrics, multiple different parameters, uh, and based on that you decide which one most uh, accurately describes the conformational changes and shifts and so on, right? So that's just for one note. And then another metric basically that we often use in your assessing the structural changes in proteins or assessing the convergence, not convergence, but large scale structural changes or compactness changes of the protein. So that is called radius of gyration, right? So radius of gyration, to calculate that, you have to first just quickly take a look at the moment of inertia. So moment of inertia is basically uh, the property of a body for which it opposes any change in its states of rest or uniform motion, right? And how you go around doing calculation for that. So let's say this one is your moment of inertia for this particular simple system here. This is just a mass m, which from a axis of rotation or from a particular axis, it is standing at a distance of r, right? So your moment of inertia will be mr squared. If there are multiple different masses which are at multiple distances from the axis, for example, r1, r2, r3, which correspond to m1, m2, and n3, then you have to do a summation of that, summation of mi ri squared. Okay. Now, if you have a moment of inertia, how you can specify your radius of gyration is that uh, so what it says is that around this specified fixed axis, uh, the radius of gyration will represent a perpendicular distance from that axis to a point where the entire mass of the body could be concentrated without altering the moment of inertia. So that means if it is a complex body that is formed of multiple different masses that are at multiple distances from a particular axis, uh, so if you can concentrate uh, your entire mass of this particular body at a particular point without altering the moment of inertia, then the distance of that particular point from the axis, that will give you the radius of gyration. Okay, So radius of gyration is oftentimes represented using this formula. So I equals m rg square. So rg by calculation will be root over of I divided by m. So that is the moment of inertia divided by the total mass of, right. And you can expand it using this formula as well. So radius of gyration is oftentimes quite important because it gives you an idea of how the compactness of your protein is changing throughout simulation. Let's see if you have a big unfolded protein, right? So a big unfolded protein, its center of mass will be located at a different point. Then if the unfolded protein is coming together, it's forming a very compact formation, right? A very rigid compact formation. So its radius of gyration will decrease because it's first of all it's a point, the moment of inertia change that will also be indicated, but the point where its mass is concentrated, kind of that point will also change, and you find out that the radius of gyration value is also changing based on different conformational changes. For example, from unfolded to folded state, the radius of gyration will decrease, right? If a protein is going from folded state to unfolded state, then the radius of gyration will increase. If a loop is sort of extending inwards of a protein, you will find out that the radius of gyration will decrease. If a loop is following simulation, it extended outward, out of the protein. So in that sense, the ROG value that will increase, right? So different sort of alterations will give rise to different changes in radius of gyration. And just as a quick demonstration of how it could be helpful, I'll just show you very briefly. Just a moment. Okay. Yeah, so what I wanted to tell you about is that how does a radius of gyration calculation help in understanding conformational differences, right? So you remember that in earlier classes, we introduced a particular software called UCSF Chimera, right? And we told that, okay, UCSF Chimera can help you in understanding conformational differences or conformational changes for simulation trajectories, let's say, right? And different types of PDB structures. You have docked protein ligand complexes. You can calculate the interactions, distances. You can identify interaction zones, right? Which residues are forming what type of bond. So those kinds of particular calculations are there. It's also a quite good visualization tool. But the main thing that we want to focus here is the visualization of a trajectory that has been generated using an MD simulation, right? So if you go to the usage of Chimera tools option, there will be MD/ensemble analysis. Okay. Now under this MD/ensemble analysis, you will find out an option for MD movie, right? So what MD movie basically does is that it will allow you to load something called a topology file. So topology files are where the structural kind of information, you can say, of the protein can be loaded. So Chimera X allows visualization of movie as well. So for Chimera X, I will have to check because um, does it allow loading of trajectory and then generation of movie? So usage of Chimera X, that I have to check actually. I don't exactly remember on the top of my head because previously I have used Chimera as well as VMT for analyzing or for visualization of movie in trajectory, but I haven't used Chimera X, so I have to check that one. Okay. But anyway, so here first of all you have to load a topology file, which since I opened up using a terminal, so you can directly browse from here. It will just go through that particular folder only. And here I have already kind of prepared the topology file, which is the complex underscore nowater.prompt. Okay. Now let's delete all of the trajectory files. You can add multiple trajectory files here, but I had a combined trajectory prepared earlier. So I'll just, let's say, use this complex underscore, sorry, this no water underscore md whole dot mdcrt. Okay, dot mdcrt is basically an extension for a trajectory file that is generated in Amber. Okay. Different other extensions are also possible, for example, .nc, .dcd, so on and so forth. But if we just press OK, you will notice that usage of Chimera gives you, of course, this particular visualization. So this seems like a protein, right? A protein where only this particular region of aspartate, let's say around threonine 39 to alanine 60. So this particular region is folded in a helix. However, this thing, the serine 1 up till alanine 37 or even proline 38, so that particular region is unfolded. It's like a loop-like structure, right? It's a loop-like structure. So whether or not you can use this structure for drug discovery, let's set that aside particularly for now. Of course, as you can see that in this structure, one drug molecule is also present, which is not binding to this structure. It's quite far away, right? Uh, if you run the simulation of it, or if you run the visualization of it, you will find out that this particular small molecule, it just moves around this space. It does not interact with this particular protein. Okay, so we'll just hide that for this current sort of loop. Okay, so I'll go to the selection and then I'll select I guess ligand, right, and I'll do action atom slash bond height. Okay, so we don't need that right now. For better visualization, again, as you know, I can color it in, let's say, dark gray, dim gray, etc. We can again go for a white background, but I guess in this case black background will also work. But if you run the simulation, so when we opened up the empty movie tab, it gave us this particular pop-up window. Okay, so here what you can do, they have also given a slider. Okay, and in the slider you see the value is from 1 to 20,000. So that means that this trajectory overall contains a total of 20,000 snapshots or frames. Okay, you can of course manually slide this particular slider, but let's say I just give it a sort of step size of 10, and then if we just run what is going on in this particular simulation trajectory, you will see that of course the loop moves around very much, right? But at some point you will see what is happening is that uh, what will happen. Okay, so let's just fast forward it, I guess. Okay, so it does not work in that way. Anyway, if you just run the movie a little bit further, you will see that in this particular position, for example, the loop is in an extended sort of conformation. Kind of, you can say that it's in a straight kind of line, right? However, the loop sort of collapses onto itself, and then it comes very close to the protein, which is the structured part of the protein, right? Let's see if you compare this particular conformation here. So now, if you compare, let's say, this position, if you remember this position, if you go back a little bit earlier, if you remember this particular position, so do you think there should be a compactness change between these conformations? So what do you think is going on as the simulation is progressing? So do you think the compactness is increasing or compactness is decreasing in this particular case? And simultaneously, do you think the radius of gyration is increasing or the radius of gyration is decreasing in this simulation? So if you go around, you will see that now the loop has collapsed. It sometimes again comes into a little bit of different conformations, but mostly it is collapsed in this simulation, right? As you see, if you compare with the earlier position, it was kind of in an extended structure, right? So that's what we got from a PDB crystal structure, let's say. But now it is collapsed. Okay. So what do you think is happening here? So compactness is increasing because it was first extended and now it's collapsing onto itself. So that means that compactness is increasing. And yes, the radius of gyration, as you have said here, the radius of gyration is decreasing. So the compactness change is kind of inversely proportional to the radius of gyration, because a lower radius of gyration will be indicative of more compact nature of the protein, right? The higher radius of gyration will be less compact nature of the protein, right? So same thing here. If you look into the radius of gyration calculation, okay, so this is the value or the data set for the radius of gyration calculation, and we will just focus on the second column. Okay, so you'll see that when the simulation first started, the radius of gyration value was around 43, right? 43 angstrom. As it goes on, you will see that it's decreasing slowly decreasing, right? So you'll see after 43, it goes to 42, then 41, 40, sometimes goes back up. But anyway, it keeps decreasing. The trend overall keeps decreasing. For example, now it's at 35, 37 range. And then if you go back again more further, you will see that it's 25, 26, then 20, 19, 18, so on and so forth. Okay. So the radius of gyration value constantly set up in a pattern of decrease, the radius of gyration changed, right? So if you take a look at what is going on via a plot visualization, you will see that if you just make it a big screen, you will see that towards the initial uh, step of the MD simulation, or towards the initial start time of the production run, it was around 45, so somewhere around 43, 44 angstrom, right? And then it gradually decreased, and then after the collapse was kind of complete, so it moved around the mean position a bit, and that's why you don't see that much fluctuation in the radiation variation at this position. Okay, so right from around 43 to 44 angstrom, it came to a value of around let's say 14 to 15 angstrom radius of gyration, right? That means it's quite increasing the compact rates, right? So if you have like a big sort of trajectory, of course, it's always useful to visualize it. But you can do what is that you can calculate the different parameters, for example, RMSD, different distances that you have a standard for a particular protein. Then also you can do radius of gyration calculations, okay, for the whole protein and for certain parts of interest of the protein, which could be useful, right, in assessing your conformational changes. So that can overall give you an idea about whether the conformational changes, basically what kind of conformational changes that you're looking at in this particular protein. For example, here, even if you didn't look at the trajectory, from just the overall ROG calculation, you could say that something is going on here, right? Either it's folding in a different way, or either the extended loop that we saw in the starting structure, that is collapsing, or some sort of conformational change is going on in this particular case. Similarly, for example, if I give you one short example, for example, in kinase proteins, which we talk about, right? In kinase proteins, if the activation segment, which is a position where the substrate binding occurs, so if the activation segment or the activation loop is oriented inwards of the protein, so that means that the substrate binding position is blocked, right? If it is oriented outward of the protein, so that means that the substrate binding position is open. Now, what you can do is you can do a radiation gyration calculation of the activation segment and kind of the surrounding region also, or you can perhaps find it out from the whole radiation gyration calculation as well. Is that if it is, let's say, increasing in the activation segment region, then of course you could probably say that the loop is changing from a folded to an extended state, right? So that will give you an kind of active to inactive, or sorry, an inactive to active conformation transition. Okay. So in this sort of way, these parameters will help you in assessing what type of changes is going on in this particular uh, set of systems, right, that you're investigating. So very useful to know about these particular parameters, and can come in very handy for assessing different stuff. Okay, so that was just very briefly about radius of gyration. But yeah, so the next set of concepts that Professor Kumiha went over were related to, for example, concepts regarding hydrophobicity, right? And this is particularly in hand related to the concept of stabilizing residues. Okay, so what is surrounding hydrophobicity? Surrounding hydrophobicity you can calculate using this particular formula, um, where so the surrounding hydrophobicity of the center residue J can be calculated using summation of Nij times Hi. Now, what is Nij? Nij is the number of residues of type I around J. So, for example, if it is the threonine that you are calculating the surrounding hydrophobicity for, it will be the different number of residues that is surrounding it. And let's say you are taking a particular cutoff of 8 angstrom in this particular case. So these residues, whichever is coming within the range, we'll calculate it only for those, right? And if you have some experimental hydrophobicity value of the residue Y, then you can plug it in directly into this formula, and that will give you the surrounding hydrophobicity. Okay. Here, for example, you have from a literature and what Professor Kumi has also mentioned in his lectures. So the surrounding hydrophobicity residues of this particular residues, right? The surrounding hydrophobicity values of these particular residues. So from residue 100 to residue 116, you can see the calculation. And based on that, of course, you can find out which residues are in buried position, which residues are more exposed to surface, uh, so on, right? So again, this will tell you the different, this will basically let you assess the conformations and the different parameters associated with this as well. Okay. Now, alongside surrounding hydrophobicity, there was also something called long range order. So earlier we had learned about long range contacts, which basically means that in the 3D space, if the residues are forming a contact, uh, and if the residues are present in a very distant position in the sequence space, so that means they are forming long range contact. So similarly, for long range order, you can plug in a formula like this. I'll just write it down as Professor Gomin also mentioned. Okay, so this is, let's say, your formula of summation of Nij divided by the number of atoms or residues, right? And if in this particular case, let's say Nij uh, will be equal to 1 if okay. So what it is basically saying that if the two residues I and J, if they are forming contact, of course, and if they are in sequence space, uh, situated at a position greater than 12, right? So there is a distance of 12 residues between those. Then the value of Nij you could plug in as 1, right? And if it is anything else, then you can plug in the value of Nij as 0. So basically, you will get the value of 1 in those cases where the residues which are distant to one another in the sequence space, they are forming contacts in the 3D space. Okay, so that one value, it will accumulate one after another, and you have to do a summation of that, and you can divide that by the total number of residues. That will give you the long range order. Okay, so again, uh, very similar calculation, but it will give you an idea about the formation of long range interactions basically. And then the next concept that Professor Gomiha also went over is stabilization center. Now, what is stabilization center? So let's take a look at the stabilization center from the original paper, one of the original papers. So it describes the stabilization centers in proteins, and it identifies characterization and predicts basically how those could be seen, or those could be calculated, right? So the stabilization centers are basically, if you look at this particular paper, you will see a definition is also given here. So here they have mentioned that two residues are considered to be long range interactions if they are separated by at least 10 residues. So they have taken a cutoff value of 10 in the sequence, and at least one of the heavy atom, uh, at least one of their heavy atom contact distances that is less than the sum of their the van der Waals radii of the two different atoms. So they have taken a slightly different approach of uh, the uh, the distance calculation, or calculation or consideration of the long range contacts. For example, they have given that one of the heavy atom contact distance should be less than the sum of the van der Waals radii of the two atoms plus 1.0 angstrom. So that is the condition they have taken. But if you classify using this way, then you will see that different residue clusters can be identified in a protein contact map. Okay, so these residue clusters will be seen where an accumulation of long range interactions will be observed. Okay, now the residue in these cores, so the cores of these clusters, they will be called as the stabilization centers. So as we know that the long range contacts, that means the distant residues are forming contacts. So likely, very likely, they will stabilize this particular fold of the protein, right? And wherever in the contact map, if you see a clump of or a cluster of sort of long range contacts present, the core of this particular clusters, they will be called as the stabilization centers. Okay, so this is because it's quite possible that in the 3D space or in the 3D structure, they have a important role in the fold of the protein. Okay, so that's how they earlier had described the stabilization center, and again quite important in the structural analysis of the protein. Okay, you can look into this paper for much more detail uh, of what they had basically talked about earlier, and you will get an idea about what is going on here. Okay, and then another concept that Professor Kumiho went over is the conservation score. So conservation score, we also earlier looked into these processes. It's basically calculation of how much the residues, certain residues could be conserved across different sequences. So again, that tells us the importance of those structural motifs or those conserved motifs of conserved residues, right? So again, quite important in assessing the functionality of certain domains or certain residues. If they are quite conserved, you will see that they're functionally relevant oftentimes, right? Okay. So Professor Komiho also went over the different methods of calculating, for example, free energy. Now, one particular method he went over was called the average assignment method. Now, based on this particular title of the method, right? It's called an average assignment method. So, of course, it is related to the calculation of averages, right? So, based on that, could you calculate this question? The answer to this question. So, here, what they have seen, what they have given us is that three different valine to alanine mutations and their associated free energy changes, right? So it's valine 12A, valine 24A, valine 25 to alanine, and valine 28 to alanine. Okay, so these four mutations have the free energy change of 1.1, 1.1, minus 0.1, and 0.3 kcal per mole. So now it's asking if in this particular protein we have a different valine residue which is situated at the 35th position. So what will be the free energy change if we mutate this particular valine to alanine? And you are asked to use the average assignment method here. Okay. Now, not very complex here. Just try to think what you can do based on the name of the method. Okay.
Sir, can you repeat the last part again? Uh, I think my connection got lost.
No, so here basically what is for this question here it is asking is that you have a protein and on different parts of the protein you have different residues present, right? For example, here we are only considering valines. Okay, at position 12 a valine was present, at position 24 a valine was present. Class um, अभी 8 बजे तक हाँ it's fine या फिर again sorry about that. Yeah, so what the question was basically saying you that the different valine to alanine mutations have been associated with some sort of free energy change, right? And it is asking you that based on average assignment, you have to calculate what would be the free energy change if we mutate a different valine residue to alanine, right? And basically, what you have to do in this case is that based on the title, of course, you know, you have to only calculate the average. Okay. So here, if you just do the calculation of the average, you will find out 0.6, right? What you can also do is that you can calculate a standard deviation also using this type of formula. We'll find out that it is coming around 0.36, so quite a large standard deviation as compared to the mean. But this could be kind of handy in some cases where you have to just get a broad sense of what goes on if you mutate a particular residue to a different one, right? And so based on this particular mutations, what do you think this 1.1 kcal per mole, for example, this valine 12 to alanine and this valine 24 to alanine mutations say? So are they favorable or are they not favorable? Just based on these values, free energy change. So since these values are in positive, right, positive 1.1 kcal per moles, so you can say
So it's unfavorable because change is higher.
yes, yes, yes. So it is unfavorable because the value is in positive, right? So the values are calculated in the way that the end value of energy is used, and that the initial value of the energy is subtracted from that, right? So end minus initial. So if the end minus initial is in positive, so that means that it was increased, the energy has been increased from an initial conformation, right? Or in the initial state. So if the energy is increased, that means it will cause a destabilization uh, in the protein. Conversely, if you had a decrease in energy, so that means that the difference in energy values that would have been in negative, uh, and that would mean that the end conformation or the end state has a uh, better sort of conformation, or has a better sort of energetically more favorable position, because reduced energy will mean that it is stabilized further, right? So in that sort of logic, you can think it out um, in the method. And here, of course, you can have. So this particular question came in another iteration of the course in the assignments, and they've given a value of 0.55 to 0.65 range. So I guess whatever value you put between these in the answer, you could have gotten it right. But you can give the value of 0.6, right, based on the average calculation, which is exactly in between this particular range. Okay. So anyway, um, now in the average assignment method, when it was calculated, so the amino acid substitutions are divided into particular beans, right? Now in this particular case, what they did is that since the mutations, for example, if you list out all of the all of the amino acids naturally occurring ones, so you will have 20 values in this particular column or in this particular axis, right? And then if you list out all of the other values, so you will get the all of the other amino acids. So you'll get particularly 19 amino acids on this axis, right? So if you divide the entire data set for the average assignment method, you don't need to write out all of the 20 times 20 400 beans. What you can do is that you could have gotten 20 times 19 beans, right? And you would have been able to list out all of the values there. Okay. So the 380 value is the total number of beans that the amino acid substitutions are divided into in this particular example. Okay. So now what are beans? Do you remember what beans usually indicate? So in any case, when you divide a data set in some way,
So
hmm,
they represent the favorable and unfavorable mutations, I guess.
no, no, no. So the beans are basically a division or a sub part of a data set. Okay. So you can think of it in a way, let's say your data set is a two-dimensional data set, right? So this is, let's say, a two-dimensional data set range. So you have some value of a reaction coordinate here, or you have some value here, parameter here that you are considering. You have some parameter here. Okay. Now within this particular data set, let's say you are trying to build, you are trying to divide your data points uh, based on easily manageable ranges. Okay, so let's say you divide your data set into this particular small square shaped or cubic shaped uh, particular range, right? And based on this, you put, okay, how many data how many data points is coming into this particular range. So this particular range will contain this many points. This particular range will contain that many points. This one is containing only one. This one containing some. This containing some, etc. and etc. Okay. So basically, your entire data set you can divide into certain range of or certain interval of values, basically. In a 1D data, you do not need to do a 2D, of course, transformation there. So in a 1D data, for example, let's say if your entire range is between 0 to 50, then you could have divided your the whole length of a range into multiple parts. For example, 0 to 2, then 2 to 4, then 4 to 6, 6 to 8, so on and so forth, right? So these small interval values, they will give you the bean. So they will be considered as one particular bean. In a 2D data set, you can divide it in this particular way, where let's say each of these boxes, they will represent a particular bin, and that bin contains some population of the data. Okay, some population, that means whichever data points were assigned to this particular range. So that means this bin's population strength is that particular value. Okay, so that's how you can divide the entire data set into bins, and then based on that you can do different types of calculations, for example free energy calculations, so on and so forth. But these are basically interval ranges uh, which you will be categorizing your data in. That's what bins are called here. Okay. Yeah. So when we discussed the PDB structures, we went over um, the B factor, right? B factor, what is it? B factor is basically a metric that calculates or that tells you about the flexibility of certain atoms, right? So if a B factor is very high, for example, let's say 50, 50, 45, 40 in those ranges, you will generally tell that this particular atom is quite highly flexible. Okay, so uh, if a residue contains all of the atoms, or let's say if a region of a protein that contains the residues, of course, and the atoms of these residues have high factor, that means this entire region of the protein that is highly flexible. Okay. Now there is another concept which is called normalized B factors. So what normalized B factors are is that you have to calculate this by B minus B mean divided by B sigma. So the B factor minus the mean value divided by the sigma value, right? So if you calculate that, what you will find out is that if the B norm value is greater than 1, then you can call it a flexible sort of atom. Okay. And then if the B norm value is less than 1, then you can call it a rigid one. So in this particular case, we will calculate this for residues, right? And in this example, what Professor Gomiha also mentioned, we'll see that a chart is given where the different stabilizing residues are listed here for this particular the different PDB codes, and then the normalized B factor value is also given on the right hand side. Okay, now you will see that the normalized B factors that are given in this particular table, they are standing at a value of less than 1. All of these cases, the values are negative, right? So that means these residues will be quite rigid, and that's why they are listed as stabilizing residues here. Because of course, if you think about it, stabilizing residues cannot be very flexible, right? So if they are themselves very flexible, so they are not taking much part in the stability of the protein. Okay, they are not taking that much part in providing the structure stability. Okay, so in this case, you can correlate that. Okay, the normalized B factors are also negative, so they are quite rigid, and that's how they are providing the stability here. Okay, so the answer to this particular question would have been false, of course, uh, because it says that positive normalized B factors are observed in stabilizing residues, which in fact is not correct. It would be that negative normalized B factors, those are mostly prominently observed in the stabilizing residues, as you can see in the table. Um, then again, you can also take a note of the earlier classes, and you can answer this particular one. So whether positive and negatively charged residues, they take a predominant part over the hydrophobic residues in stabilizing a protein. So that means that what it is talking about is that in the overall fold or the overall stability of the protein, whether the contribution of hydrophobic residues is lesser, much lesser than the positive or negative charged residues. Okay, so should that be true or false? Yeah, as you say in the chat, it should be false, right? Because predominantly what drives the fold of the protein and what drives the stability of the protein, those are hydrophobic interactions, right? The hydrophobic collapse is what drives the hydrophobic clustering is what drives the folding, right? And in this particular case also, if I take a reference of this paper, you will see that it says in their analysis that more than 80% halophilic, thermophilic, alkalophilic acidic proteins, they showed a trend of having a higher amount of glycine, alanine, isoleucine. Okay. Then alkaline, cyclophilic, halophilic proteins showed a higher amount of isoleucine in their sequence. If you notice, all of these, they are classified under non-polar residues, right? So that means that these hydrophobic or non-polar residues, they are quite important uh, in the 3D fold of the protein, because their prominence is again, you see, very high in those cases, right? And if you want to remember what the classification of proteins looked like, so it looked something like this, where your glycine, alanine, proline, valine, leucine, isoleucine, methionine, those are kind of classified as non-polar. Okay. So that was one set of concepts. And then another one set of concepts Professor Gomiha went over related with the stability and mutations and mutation-induced changes in stability or distability, right? So in this particular case, what do you think? In buried mutations, which properly will be correlated the most with stability? So again, since the term buried is coming here, what do you think is more correlated with that term among the following options? Yeah, hydrophobicity, as mentioned in the chat, right? So hydrophobicity is what will be the changing the most, and what will be the what is sort of expected to have the most correlation with stability in buried mutations, because again, buried versus surface exposed, so they have a, of course, difference in the amino acid constitutions, right? The buried interfaces or the buried regions will have hydrophobic residues. The surface exposed regions will have the residues which are not hydrophobic, right? Polar residues. So that means that the stability in this case will be more attributed to the change in hydrophobicity. Okay. Now, what kind of changes following mutations can occur in protein? Of course, the change can cause stability of the protein, stability of certain regions of the protein. The change can also destabilize the protein. The mutation can also destabilize the protein, right? So in this case, what the question is asking is that which of the following mutations do you think will be highly destabilizing in proteins? So is it tyrosine to alanine, valine to histidine, glutamic acid to methionine, or isoleucine to lysine? Now, this particular one, you might try to figure it out using the particular sort of characteristics of the amino acids that are mentioned here, of course. But Professor Gomiha, he also showed an example of a chart where it's quite long time ago. The paper, I guess, that is referencing it. But what they calculated is that uh, they had measured or computed the frequency of destabilizing and stabilizing mutants uh, upon denaturation. Okay, so what it is saying is that in the left hand side column, you will see the initial sort of residues. Let's say before mutation, what residue was present, that is mentioned. And on the other hand, you will see that which residue it was mutated to, that is mentioned. Okay, and if you read what is written here on the bottom, so it says that the mutation is, of course, from left to right, and the frequency of stabilizing mutations are shown in parentheses. Okay, so out of all of the mutations, I guess, how many mutations were stabilizing? Those are mentioned inside parentheses. And if you look at the Y2 alanine value, 22 is outside parentheses. So that means out of 22 mutations, only one is found to be stabilizing. So that's quite a high difference, right? So the number of stabilizing mutations that they found was much lower than the destabilizing ones. So that means that these are highly destabilizing. I guess the value that is outside the parenthesis, that could probably be how many mutations they found where uh, it caused a destabilization in the protein structure. Okay, not the total number of mutations that they have found. But anyway, so let's say it is destabilizing versus stabilizing value for outside and inside of the parenthesis, and this one is standing out the most because it is indeed the most destabilizing mutation here. Okay, so from this one, if you calculate the other options, so valine to histidine, we'll find out this one. So one destabilizing, zero stabilizing kind of, right? E2M, you can also see uh, in this value, I believe. Yeah, in this one. So again, it's 0, 1, and isoleucine to lysine. So isoleucine to lysine, which is 0, 0. So they didn't find any sort of changes in this pattern, okay, destabilizing or stabilizing. But anyway, so in what ways do you think that stability or distability can occur? Okay, so here it is asking that if hydrophobic residues are crowded in the core and alanine is mutated to valine, what will happen to the structure? Now, this is giving you, of course, the mutation, but also the context for the mutation. The context in this case is the surrounding environment. Okay. So what it is asking is that firstly, you have a crowded environment. So crowded environment means not a lot of space to move uh, in this particular environment, right? Not a lot of space to move for the residue, and not a lot of space where if some change is occurring in the residue, which is leading to a bigger structure, that could be adjusted. So there is not a lot of space for that. Okay. Now, in this particular case, you are saying that alanine is being mutated to a valine residue. So what could happen here in this case? Yeah, I see create steric hindrances, right? So it will, of course, create steric hindrances because if you look at the size of the residues, so you will see that the R group for alanine is CH3, right? But in this case of valine, you have CH, CH3, CH3. Okay, so it's a quite bigger, bulkier group. And what that will create is that if you suddenly change this alanine, which was nicely packed in the structure, if you change it to valine, if you try to change it to valine, due to the bulky nature of the side chain, so it will create steric hindrances. Okay, so that will be uh, a stabilizing or destabilizing mutation in this case. So it will be a destabilizing mutation because steric hindrances are being created here. Now, similar sort of mutations could also be stabilizing if, for example, it had changed from a bulkier residue to a smaller residue, but the adjustment or the fold of the particular protein was quite well adjusted. The spaces and the gaps were quite well adjusted in the mutation or in the mutated state, right? So look at the examples here, particularly. So if you have a negative to positive interaction, a charge-charge interaction, right? So already a Coulombic interaction was present, which was giving the structure stability. Now, if you change the positive charge to a negative charge, so that means that instead of attractive force, now they have a repulsive force between them, right? So, if this particular interaction was important for the stability of the protein, so now, of course, the protein will be highly destabilized. Okay, that is one destabilizing mutation. And in the next case, what you have here is that you had these residues, right? And this red one was mutated to the green one. So this will cause also a change in the stability, right? And in the other one, you will see that uh, this is happening where it was already closely packed, but now it's due to the change into a bulkier kind of structure, it is having some sort of steric clashes. So among all of this, which ones will be stabilizing and which ones will be destabilizing? So are there any stabilizing mutations at all in this example? We already know that positive negative charge, so this one is not it, this one will be destabilizing, right? Because in initial case there was a charge interaction, now you have a repulsion, so that's destabilizing it most likely. uh, in the next case, however, it will be a stabilizing sort of mutation in a general sense because it is a space filling mutation. So it's saying that already there are some gap present in the structure which could accommodate a more bulkier residue, right? A bulkier residue. And now, since the space has been filled up following mutation, so there will be a better packing. So a more a tighter packing, right? So that will most likely indicate that it will stabilize the structure because of a tighter pack. And of course, the next one will be destabilizing because due to steric hindrances, it will very much destroy the structure or the fold in that region, right? So that was one thing. Um, Now, based on this kind of informations, and as you can know, as you know, that we always like to convert these informations into energy changes, right? So, based on that, again, this question is trying to find out something. The answer to this question is letting you try to find out something. Okay. So here it is talking about different alanine to valine mutations. Okay, 1, 2, 3, 4, and they have associated free energy changes also. All are in kcal per mole. the changes have been given here. Now it is trying to ask you that find out the most tightly packed residue position before mutation. Now here is a convention change. So we always know that a positive energy will mean uh, destabilizing generally, right? Final minus initial, that is positive, so that is higher in nature, so it had an energy gain, so that means it will be destabilizing. But in this particular case, the notation is slightly different. So they are saying that the positive mutations at the positive energy values, that is stabilizing in nature, and the negative energy values, that is destabilizing in nature. Okay, so let's just take into consideration this notation only right now, and based on that, could you tell me which of these free energy, which of these mutations, considering all these mutations, which particular alanine was in a position of tighter packing before mutation, before changing to valid? Just remember that positive is stabilizing mutation here and negative is destabilizing mutation. So I see one answer D in the chat. So D is telling you about A58 to V, right? Alanine 58 to valine, and the associated free energy change is 3.2. Okay, so 3.2 here in this case, the positive value is stabilizing, right? So that means that alanine 58 to valine mutation is highly stabilizing. Okay, now if alanine 58 to valine mutation is highly stabilizing, do you expect that the alanine was situated in a tightly packed position? Because remember, if it was already in a tightly packed position, then changing it to valine with a more bulkier side chain, that is expected to destabilize the protein, right? But in this case, it is of a positive value, which is denoted as stabilizing. So that should not be the case here. So what you have to find out here is basically um, the value that was uh, destabilizing, the most destabilizing following mutation. Because if in the earlier situation, it already had a tighter packing, then if you increase the side chain bulk, of course, it will lead to a steric hindrance and it will cause destabilization, right? So the more tighter packing earlier it had, the more destabilized will the mutation be. Okay, so that means that since destabilization is indicated via negative term, so your most negative value, which is minus 2.4 kcal per mole, corresponding to alanine 42V, so that will indicate the position of the alanine that had the most tight packing. Okay, so in this case, it will be A42 position, because if you change A42 to valine, it causes the most destabilization. So that sort of logic is occurring. Okay. Yeah. So here again, the question was asking that the mutants were divided into 380 potential substitutions, and the stability of each mutation was assigned using data from similar types of mutations. Of course, we know that this is the average assignment method, right? 20 times 19 matrix or beans, you can think of it. Okay. So that was one thing. And then just for information, so van der Waals interaction is quite important when you try to assess, for example, protein drug interaction, etc. And usually it is calculated via this Lennard-Jones potential. Okay, so just remember this particular formula. You don't need to know all of these terms or remember all of these terms, but the plot kind of looks like this, and this is the formula by which you can calculate a Lennard-Jones potential parameter. Okay. So again, some other concepts related to mutations we can go through. For example, in exposed coil mutations, which of the following can be correct? here, as you say, the answer is C in the chat. So there will be inverse relationship between hydrophobicity uh, and stability, right? So you can again characterize the type of mutations, the type of change in the characteristic of the mutations, for example, hydrophobicity, and then what could be the relationship in those sense. Okay. So the explanation is kind of here. So it is buried mutation. If it is buried mutation, so in that case, the stability has a positive correlation with hydrophobicity. Okay. So of course, in a buried sort of environment, so if the mutated residues are also becoming hydrophobic, so if it is a change from towards a hydrophobic residue, so that means that the stability will become higher because these sort of residues, hydrophobic residues, prefer a buried environment. Whereas in the case of exposed mutations, exposed coil mutations, uh, if the residue is changed to a hydrophobic residue, so that will not like this sort of solvent exposed environment, right? So that mutation will be destabilizing in that case. So it has an inverse or negative correlation with stability. So in that logic, you can previous question means this one, exposed coil mutation, or this one. So this is just the formula for Lennard-Jones potential. Okay. So it's calculated using this term. So there is an attractive term, there is a repulsive term. And based on those, you can find out the Lennard-Jones potential calculation. If we're talking about this one, this is the average assignment Bean method. So we also looked at it earlier. And in this particular case, of course, the buried mutations will be having positive correlation with hydrophobicity because of the nature of the residues. So hydrophobic residues will be favored here in a buried state or a buried environment, that will have a positive correlation, and in exposed kind of environment where the hydrophobic residues will very much not like the environment. So that is why the stability will have a negative correlation with the hydrophobicity. Now here again the question was asking the performance of a computational method for discriminating, stabilizing and destabilizing mutation. What does it happen to that when the dataset is classified based on secondary structure and solvent accessibility? So of course, if you have a dataset classification, the more nuanced the classification is, the better your computational algorithm will be performing on that, right? So in this case, if multiple criteria are included, for example, secondary structure, solvent accessibility, and if some other parameters also included, so that will give you more nuance basically, right? So your data set, if it is classified based on those, so you can say that the algorithm will be better able to identify destabilizing and stabilizing mutations. Okay. So that is the sort of mutation related concepts that Professor Promeha went over. So in some of the next classes, we will also look into the computational methods of drug discovery, docking, virtual screening, and simulations in more detail, which will give you a kind of an idea about how you can use it in your own research or how to proceed in the protocol one after another, right? So yeah, so that is, I guess, all that I will have for this particular session. Hopefully, you learned some how to solve some questions, and hopefully I was able to revise some of the concepts that Professor Kumiha mentioned. And then yeah, I think for today I'll just wrap up, and then I'll see you all. So again, very very thank you to all of you for joining.
Thank you so much, sir.
Yeah, and then we'll try to meet on next. So next week might not be possible. So I'll try to request the NPTEL platform for rescheduling the classes because I'll be not available for the conference. But yeah, so that will be notified by the NPTEL team to you in email. Okay. So thank you all of you for
Okay,
joining.
sir.
Yeah, and then we'll meet in the next class. Okay.